Students, good morning again. We're going to look at the development of arteries and veins in a PowerPoint manner, and then I'm going to take you through the development of the lymphatics differently. Now, I want you to appreciate certain type of tissues. The first set of tissues that I want you to appreciate are called the angioblastic tissues. The earliest blood vessels are derived from the angioblastic tissues, which differentiate from the mesenchyme in three regions. Now, mention these three regions: the surface of the yolk sac, the body stalk, and the chorion. Also, it is important at this stage for you to understand the meaning of three words: vasculogenesis, angiogenesis, and arteriogenesis. We're going to look at vasculogenesis first. This is the process of blood vessel formation occurring by de novo, from the beginning, production of endothelial cells. So it is the formation of blood vessels from endothelial cells. Okay? From the beginning. And it is different from angiogenesis, which I'm going to talk to you about now. Angiogenesis denotes the formation of new blood vessels from pre-existing ones. So the one that was formed from vasculogenesis, you can get angiogenesis from it. And you appreciate the difference between angiogenesis and vasculogenesis, which is the formation of new blood vessels from pre-existing ones. That is angiogenesis. Whereas vasculogenesis is the term used for the formation of new blood vessels from the endothelial cells when there were no pre-existing ones. So that's why I said from the beginning. There are two types of angiogenesis: the sprouting angiogenesis and the intussusceptive angiogenesis. The formation of medium-sized blood vessels, the tunica media plus adventitia, is reserved for arteriogenesis. So you now know angiogenesis, vasculogenesis, and arteriogenesis. And let me tell you, as far as I am concerned, this is all you need in this lecture, the terminologies. And I can tell you, you can test some of your senior colleagues who are now doctors and dentists and ask them about angiogenesis, arteriogenesis, and vasculogenesis. They can't define it. They mix it up or they don't know it. So you need to know that the formation of medium-sized blood vessels, tunica media plus adventitia, is arteriogenesis. And angiogenesis denotes the formation of new blood vessels from pre-existing ones, whereas vasculogenesis is the term used for formation of new blood vessels from the endothelium when there is no pre-existing one. Let us look at what happens in the third week of intrauterine life. In the yolk sac and the base of the body stalk, small, more or less spherical group of cells are found early in the third week. These cells are called the blood islands. You can see them here. You can see the developing endothelium. You can see the neuroblast early stage. You can see the hemocytoblast. And this illustration is taken from Gray's Anatomy, and it's a section through the wall of the yolk sac of an early human embryo to show an early stage of the differentiation of angioblastic tissue. And similarly, this illustration is showing the endothelium and the blood island, which contains the normoblast. We're going to now look at the formation of vascular endothelium. The stages of transformation of blood islands into blood-containing vessels have not yet been demonstrated in detail, but it is generally believed that the peripheral cells of the island become flattened and form vascular endothelium. The central cells becomes converted into primitive red blood corpuscles. Later, this small blood-containing space form a continuous network of small vessels. In the chorionic end of the body stalk and the mesoderm lining the chorion, typical blood islands are not found, but the cells of the mesoderm give rise to solid strand of angioblast. With regards to the blood vessel formation, each strand contains 2 or 3 rod-shaped nuclei arranged in a single row and soon comes to contain a space occupied by 1 or more nucleated hemoglobin-containing cells. The space will now coalesce, come together to form blood vessels, which are therefore lined by derivative of the mesoderm. There is a little uncertainty, a red area. The precise source of these contained blood cells is uncertain. The earliest blood vessels are formed at several centers. From the walls of these vessels, buds grow out, become canalized, converted into new vessels, and join with those of neighboring area to form a closed meshwork. The heart and the blood vessels of the embryo arise from angioblastic tissues differentiated from the intraembryonic mesoderm. That is one point that I want you to appreciate. Secondly, prior to the establishment of the circulation, new vessels develop in situ, but thereafter they all take origin as outgrowth of pre-existing vessels. So, what I want you to do is to study this complex illustration. One side is induction and the other side is resolution. It is down regulation of proliferation and migration and reformation of the basement membrane, okay? Vascular basement membrane, all right? So this will now continue shortly as the development of the lymphatics. And the reason is the lymphatics form differently from the veins and arteries. All right. While the blood vessels begin to develop around the end of the third week, the lymphatic system begins to develop at the end of the fifth week. Approximately 2 weeks later than the cardiovascular system, that is the blood vessels. One view states that the lymphatics develop as diverticular of the endothelium of veins, whereas another states that I'll put it this way. One view states that the lymphatics develop as a diverticular of the endothelium of veins, whereas another states that blood-like vessels develop from clefts in the mesenchyme that connects the venous system secondary. The cells lining the mesenchymal cleft assume an endothelial shape, and subsequently sprouting of these cells causes the cleft to fuse and form the lymphatic channels. And the truth basically is, one is not so certain if you need all this information. So I'm trying to gather this properly for you. Okay, so that's it. So in week 6 and 9, local dilatations of the lymphatic channel form 6 primary lymph sacs. 2 jugular lymph sacs near the junction of the subclavian vein with the anterior cardinal, which becomes the internal jugular vein. And then you have 2 iliac lymph sacs near the junction of the iliac vessels with the posterior cardinal vein. And then 1 retroperitoneal lymph sac in the root of the mesentery on the posterior abdominal wall. 1 so-called cisterna chyli, which you must have been told about in your lecture on lymphatics, which is dorsal to the retroperitoneal sac at the level of the adrenal glands. This is just for your information. I don't think anybody's going to ask you about these details in an exam. Now, lymph vessels grow out from the lymph sacs, which I mentioned, along with the major vessels to the head and neck, the arms, and the arms from the jugular sac to the lower trunk and legs from the iliac sac, and to the guts from the retroperitoneal and the cisternal sac. So for the head and neck and arms you're looking at the jugular sac. For the lower trunk and leg, you're looking at the iliac sac. And for the gut, you're looking at the retroperitoneal, peritoneal, and the cisternal sac. The cisternal chili is connected to the jugular lymph sac by two large canals, the right and left thoracic duct. An anastomosis forms between the two ducts, thus the definitive thoracic duct is formed by the cordial portion of the right thoracic duct, the anastomosis, and the cranial portion of the left thoracic duct. The right lymphatic duct is derived from the cranial part of the right thoracic duct, and both the right and left thoracic duct join the venous system at the angle of the subclavian and the internal jugular vein at the base of the neck. And what I just want to do additionally is to talk about the development of the lymph nodes, except for the upper portion of the cisternal chili, which persists. The lymph sacs are transformed into group of lymph nodes. So you see what the lymph sac does. It forms both the lymph vessels and the nodes. And this is during the early fetal life at about the 3rd month. So the surrounding mesenchymal cells will invade each of the sac and break it up into lymphatic channels or sinuses. The mesenchymal cells give rise to lymph node capsules and connective tissue framework of the nodes. The lymphocytes seen in the node before birth come from the thymus gland. The lymph vessels and the germinal centers of the lymphocytes production do not appear in the node until just before or after birth. And finally, the lymph vessels also develop along the course of other vessels. And I'll be doing a grand revision for all of you next week or this weekend, and I will send the link to you. And you will have an opportunity to ask me questions on the development of the blood vessels. You'll have the opportunity to ask me questions on glossopharyngeal nerve, vagus nerve, accessory nerve, and hypoglossal nerve, and also you'll be able to ask me questions on visual pathways. Thank you, ladies and gentlemen.